I think there's immense value in having integrated optogenetics and imaging. nVoke for us came at exactly the right time. We had a hypothesis that a particular class of interneurons was regulating calcium signaling and neuroplasticity in a particular cell type. But we had no way of actually showing this during the task that we were studying. And what nVoke allowed us to do was actually inhibit these interneurons while imaging these calcium events that we cared about in the context of this task. And for us, that was absolutely instrumental to the success of the project.
In vivo calcium imaging allows us to determine the neural correlates of behavior. And when we add in simultaneous optogenetics, we can then begin to investigate the causal link between neural circuits and behavior. In addition, we can investigate how defined populations of neurons are controlled and regulated by other populations of neurons, both within a local microcircuit as well as within interconnected brain regions.
In the initial design stages for nVoke, we really had to consider the crosstalk issues between the opsin and the calcium indicator, as well as optical crosstalk, which can occur within the microscope itself. Larger microscope systems at a benchtop scale are much better suited for rejecting stray light than a system of this size. We had to rely on very careful design of the optical filters in order to eliminate stray light caused by this very powerful optogenetic light source that's being paired with the calcium imaging. The nVoke system is far more than just a device. We've done our work to design a device which performs impeccably, but on top of that we provide support for the user in the entire workflow from the surgery all the way through data analysis.
Neuroscience is sort of undergoing a revolution. The field is really moving towards this integrated approach where you need to really do all these things at the same time and really put together, how do circuit elements impact network dynamics, how does that in turn impact behavior? And to do that, you need to be able to measure these things and perturb these various elements, at the same time, while the animal is performing these behaviors. And what nVoke has allowed us to do is essentially achieve this goal of simultaneous perturbation, imaging, and freely moving behavior.